Hello everyone, my name is Florin Walter. I'm a PhD student at the European Molecular Biology Laboratory and also affiliated with the German Cancer Research Center. In this talk, I will present my latest work together with Oliver Stegl and Britta Felten, which has the title FISHFactor, a probabilistic factor model for spatial transcriptomics data with subcellular resolution. Many important aspects of biological systems, such as tissue architecture or cell-cell communication, can be studied by observing the spatial locations of RNA molecules. For this, a number of spatial transcriptomics techniques have been developed, which can be roughly classified in two groups. Techniques in the first group are based on next-generation sequencing coupled with spatial barcodes. They provide molecule counts for a set of genes in a set of spatial regions. This way, they generate data matrices with samples corresponding to spatial regions and features corresponding to genes. However, the spatial regions are typically larger than single cells, and the resolution therefore is at most at the cellular level. Techniques in the second group are based on in situ sequencing or in situ hybridization. They provide exact spatial coordinates of individual RNA molecules, thereby generating coordinate lists with gene labels. Because of the high resolution, these techniques may be used to study subcellular gene expression patterns, which are essential to further advance the understanding of cell heterogeneity beyond the cellular level. Vector models are a commonly used method for analysis and dimensionality reduction of high-throughput datasets in molecular biology. The basic idea is to decompose a high-dimensional data matrix into the product of two lower-dimensional matrices, called weights and factors. Formulated in a Bayesian framework, vector models offer the flexibility to use different priors and observation models to account for different prior knowledge and data types. Several factor models have been proposed for spatial transcriptomics data at the cellular level. The baseline model is factor analysis with a link function and a Poisson likelihood to account for the count nature of the data. Such a model has univariate priors on the factors and thereby assumes independent samples. This is, however, not necessarily appropriate for spatial data, because, for example, gene expression profiles are expected to be more similar at points that are close than at points that are far apart. It has therefore been suggested to replace the univariate priors on factors with Gaussian process priors to model spatial covariation and obtain spatially smooth factors. This model with spatial factors has been further extended by constraining the weights and factors to the non-negative range, to obtain a part space factorization that increases sparsity and improves interpretability. However, all these models require data matrices as input and are therefore not directly applicable to single molecule data, which comes as a list of coordinates with gene labels. To apply the models to single molecule data anyway, there are two options. One option is to aggregate coordinates in spatial bins and thereby obtain a count matrix, as required by the model. However, this has the disadvantages that in every case some of the spatial information is lost and that a binning resolution needs to be chosen manually. A better option is to model the molecule coordinates directly using a spatial Poisson point process observation model. This stochastic process has a probability density that involves an intensity function lambda, which has high values where many molecules are expected, and vice versa. A Poisson point process likelihood has been used before in factor analysis applied to temporal data in neuroscience. We propose a probabilistic model called FISHFactor, which combines the benefits of the existing factor models, shown before, with an observation model suitable for subcellular spatial transcriptomics data with single molecule resolution. The input to this model is given by a list of RNA molecule coordinates with gene labels in a previously segmented cell. This is illustrated on the left side of the figure with the complete cell on the left and the same cell divided by genes on the right. For every gene, the molecule coordinates are directly modeled by an independent spatial Poisson point process, which is characterized by an intensity function as shown on the right of the coordinates. The intensity functions for all genes are then decomposed into the product of a non-negative weight matrix and a small number of non-negative spatial factors. These factors use Gaussian process priors with a Matern kernel to promote spatial smoothness. Finally, FISHFactor allows to model multiple cells jointly by inferring a single weight matrix that is shared between cells, but using independent factors for different cells. This way, it is guaranteed that factors have the same interpretation across cells, and that it is possible to dissect cell-cell heterogeneity by considering variation in factor activity across cells. FISHFactor is implemented using stochastic variational inference and sparse variational Gaussian processes with the probabilistic programming language Pyro and the Gaussian process package gPyTorch. In our first experiment, we simulated data in a way similar to the generative model of FISHFactor. We generated various datasets with different average numbers of molecules per gene and per cell, as this also varies in real datasets. This is illustrated here using different colors, where blue denotes the lowest average count of 17 molecules per gene and cell. We applied FISHFactor to this data and correlated the inferred weights and factors with the simulated counterparts. As expected, the construction was more accurate with a higher average number of molecules per gene and cell. To compare FISHFactor to an existing method, we performed spatial binning with various resolutions on the coordinates and applied non-negative matrix factorization. By correlating inferred and simulated values, we found that FISHFactor more accurately recovers the factors in all datasets and more accurately recovers the weights, especially in datasets with a low average number of molecules. This suggests that the advantage of using a Poisson point process likelihood over spatial binning becomes especially apparent when the average count of molecules per gene and cell is low. In a second experiment, we simulated multiple cells with a shared weight matrix and compared the reconstruction accuracy of a single FISHFactor model applied to all cells jointly with independent FISHFactor models applied to the cells individually. We could show that the reconstruction of weights benefits greatly from modeling cells jointly, while the factor reconstruction is also slightly improved, even though the factors themselves remain independent. Finally, we applied FISHFactor to a real dataset of 225 cultured mouse embryonic fibroblast cells from the SecFISH+ paper. In the original publication, 60 genes with meaningful subcellular spatial expression patterns were identified. We applied FISHFactor to the same 60 genes in all 225 cells. We found in the inferred weight matrix that most genes have a high weight on a single factor, revealing 3 gene clusters. Looking at the factors, we also found that one factor captures genes that are expressed at the cell border, one factor genes that are expressed around the center, and one factor genes that are expressed in the cell center. This is also in accordance with the findings from the original publication. Moreover, we used the scale parameter of the Gaussian process kernels of the factors to perform cell clustering based on variation in factor activity across cells. To conclude, we developed a probabilistic factor model that can be applied to single molecule resolved spatial transcriptomics data to reveal subcellular spatial expression patterns. We showed how FISHFactor outperforms a binning-based approach and applied it to a first real dataset. With this, I want to end my talk and thank my collaborators and supervisors Britta Felten and Oliver Stekle. We have a preprint of FISHFactor online, which will also get an update in the coming weeks.